Yes.
Yes, yes.
Is Muslim back? I think he's back, right?
Yes, yes. No.
So he should be here, Melanie and
Map.
Matt. Hey, Muslim, welcome back.
Hello, how are you?
Good. How was the neuroscience meeting?
It was a good experience.
It was in DC.
Yeah. How are the presentations going on? How are the presentations going on? Slightly cold out there.
Slightly.
Slightly.
Pretty cold there.
Yeah. I don't think cold is a problem. Like the wind is more of a
Yes,
problem.
it's the wind for sure.
Wind.
It becomes difficult when it's windy. And for how long are you in Pittsburgh?
Me?
Yeah.
Since, hi Melanie, yeah, 2002,
23.
21 years.
So this cold is also harsh for Pittsburgh, or it's normal
no.
winter cold?
It's harsh for December or November. Usually this is like January, more like end of December, January.
Because everyone said last year that it was the mildest winter.
Yeah, I hear this. I don't know. Yeah, I don't know. I haven't looked. But yeah. This is unusual for November. This is pretty cold for November. But not unusual for winters here. Not really. Yeah, 20s and 30s. Usually in January. So I told Oksana I need to leave at 9:45 because I have a thesis defense that I was the only holdout, and this is like the only way I could do it was to cut this class a little bit short. I'm also missing another meeting on the other end of it. But anyway, so I have to leave at 9:45. So Oksana said she thinks she can get through her. So it's just her thing. Yeah. You have Melanie. So Melanie, you'll be first on Thursday. And then Moonsong, next. Matt's next, and then you.
So I'll be presenting on Tuesday.
Probably Tuesday, yeah.
Or it's going one presentation in a class, or
No, we did two the last time. No?
Just one at a time.
We did one at a time. Well, on Thursday we're gonna have to do two somehow.
So I'm on Tuesday, not like on
Right, Tuesday. You'll be Tuesday.
So it's me and Matt.
Yes.
Slight mistake here. So
It's okay.
Get this overlook things. That would be great.
No worries.
You are quite right about the SFN. There were 25,000 people over there.
How many?
25,000.
Yeah.
You said that
I
just
told
don't be depressed.
Right? You see all these people, you're like, how am I gonna make? Yeah, it's a little depressing the first one.
Yeah, like so many people working in same direction.
Yes.
Same field.
Same field, same things.
But amazing thing is, even then we cannot know much about the brain.
I know, right?
It's so complex.
It's so complex. I know. It's amazing. Yeah, it is amazing.
Every time I think it's a lot out to research
Yes,
in that field. Like,
right.
yeah.
I know. I know. The brain is amazing. I miss it. I miss studying the brain. I need to go back to it a little bit. Cancer's kind of like that though too, because every time we think we know something, like I don't know, cancer's smart, unfortunately.
Ma'am, do you think that we are going in the right direction for research of cancer?
So you're
It is a good question. I mean, yes and no. I think looking for targets, like specific targets to decrease toxicity of chemotherapeutics is a good idea, but I don't know that anything's going to be as effective as chemotherapeutics is the problem. You know what I mean? Because cancer's tough
Yes.
and you need to kill it. So
But you kill a lot of normal cells.
I know. So I don't know, and I think the using the immune system is smart, like trying to get like, but then I think what's going to happen. This is just what I think. I think cancer cells are going to be smarter, and they're going to start to
Right.
mutate and
Yeah.
change, and
Just like
I think that's
bacteria.
why we're never going to really cure cancer, unfortunately. Yeah, unfortunately.
You're smarter than the whole human organism.
Yes, right. These cells, these stupid cells are so. And it gets me so frustrated when I hear people in the lay public say that, oh, we have a cure for cancer. It's a money maker. So I mean, I see where it's coming from, because pharmaceutical companies can be crooked, obviously, and do make a lot more money than necessary.
Yeah.
But I don't think anyone's holding back a cure for cancer. So.
Why their cells shouldn't be smarter? Like, we cannot compete with the bacteria, which is not even eukaryotic.
I know,
They
right?
can be resistant a lot. Like,
Yeah,
they are updating themselves
yeah,
quite frequently.
Right.
Like, they are beating us every year. Like,
yeah, yeah.
So, a human cell, a cancer human cell, is better than the bacteria.
Yeah, right,
So,
yeah.
we have more resources.
Mhm.
It's
Yeah.
difficult.
It is difficult. I do think personalized medicine will help though too, like understanding everyone's, as if we can understand everyone's tumor to a point where we can like put it into some computer modeling maybe and come up with the best treatment regimen for that particular person, but that's very expensive, you know, and I don't know if that's even, you know, I think that'll be helpful, but I don't think that'll be perfect.
And also not hard and soon. Not very
What's
soon.
that? And it's not gonna happen very soon, no.
But it's a good idea about
Yeah.
like
I think every
putting
person has to have an individual regimen almost. I don't think we can just be like, this is this.
Yes,
you
yes.
know?
because there's a lot of
Yes.
variability among the cancer cells,
Yes.
even in the same type of cancer. Even
Yes.
like, have you guys have seen in the breast cancer?
Yes,
There are many types of breast cancer.
right.
So
I
it's
mean, we
not
have
like,
3 main categories,
Yeah.
but then when you look at triple negative, for example, there's, I mean, so many variations of triple negative.
yeah.
And I'm sure ones we don't even know about. So. Okay.
Sorry,
Sorry, that's not good. It's a very interesting
No, it
conversation.
was a
Sorry.
good conversation.
No, I like these conversations.
So,
Okay.
is it okay if I just sit and then
No,
present?
yeah, you can sit.
Because
Yeah.
That way it's easier to control.
Yeah, no problem.
So good morning and welcome everyone for my presentation. So me and Melanie will be presenting chapter 9. I will be presenting protein kinases A and she will be presenting C. So before I dive into protein kinase A or PKA, I will go briefly about what are the kinases, protein kinases, and then we will jump into PKA and what it does to our biological system, and lastly I will briefly describe a drug that targets that enzyme. So kinases are enzymes that act as a catalyst, and it transfers one phosphate group from ATP to a substrate, which is usually protein. The name kinase came from Greek origin, which means to move, since it moves phosphate to protein, hence the kinase. And PKA is protein kinase because it targets protein. So it's very ubiquitous in nature. So one scientist found in one single cell of Saccharomyces pombe 120 protein kinases, and that's just an yeast cell. And in human till date the book was published, there was 518 protein kinases was discovered. Now, what are the kinases? Kinases are basically a switch in our cell. So it can turn on an inactive protein into an active stage and then start a reaction. On the other hand, it can turn off an active protein and stop a reaction. But most of the time it is related to the activation, and going forward in this presentation I will be referring mostly to the activation activity of the pKa, but know that it can also turn off an active state of protein. And in this figure you can also see a phosphatase. I will discuss that later also. So kinase is a switch in cellular reaction. And in the book there are several other kinases that they discussed, but for this presentation I'll be focusing on pKa only. So what is this phosphate group that the kinase transfers and the reaction happens, or activation inactivation happens? So as I mentioned that to perform its activity, pKa needs ATP to take the phosphate group out of the ATP and transfer it to a protein. So this is the structure of phosphate group and this is the structure of ATP. Now, as you can see, this adenosine triphosphate, this triphosphate moiety is like very high energy binding. It means that it has very high energy, and as we know in nature, something that is very high energy will always have a tendency to break down, release energy, and acquire a lower state. So pK use this bond, like this two red marked bond, which is very high energy bond. So it cleaves this phosphate group from ATP, converts it into ADP, a lower energy state, and then transfer this phosphate group to another protein. When this phosphate group binds to the substrate, which is protein, it causes an allosteric changes in that protein moiety, which will start our end-end reaction. So this is what this is the function of pKa that it usually initiates. It initiates binding, recognition, activation, inactivation. So from phosphate group, we have to discuss about the phosphatase, because in the previous slide I mentioned that pKa is a switch. So like any switch, when you came into this room, we turn on the light, and when our class is over, we'll turn off and go away. So on and off is like a balance in nature. So is in the cell. So when pKa turned on a protein by adding a phosphate group, and it will perform its activity, give its signals, it has to be turned off and then bring back to the inactive stage after the reaction. So for that we need phosphatase enzyme, and we cannot discuss kinesism without the phosphatase. These two are the balance in nature. So this is the substrate. After pKa binds a phosphate group to a substrate, it will give a signal out, and then phosphatase, with the help of a water molecule, will again convert the substrate into the inactive stage and release the phosphate, and it will bind to another ATP molecule and convert into ATP. So this is the balance in the cell. If any of the process, like if the kinase is hyperactive or the phosphatase is overactive or underactive, anything that disturbs this balance will cause pathological condition. So for maintaining a balance in the cell, both kinases and phosphatases needs to be working in sync. So I will be discussing protein kinase A. This is a three-dimensional structure of a protein kinase A. This is the inactive stage. As you can see, they are color coded, like two different parts. One is pink and one is blue. What happened is protein kinase A is activated by CAMP, intracellular concentration of cyclic AMP decides whether pKa will remain in inactive stage or in active stage. So when CAMP binds with pKa, it changes its conformation or its structure and release the active moiety that will then go on and bind a phosphate group to a protein. I will discuss that in details in the next slide. So this is just a general idea how pKa exists. It's kind of a tetramer. As you can see, two different active sites and two different inactive sites. Not inactive, like CAM binding site. So when CAM binds with this blue portion, the active moiety, this dimer is released and they perform the phosphate binding activity. Now PKA does a lot of works in our cell. Its short-term regulation is like it controls the glycogen metabolism from glycogen to glucose metabolism, and it also does like a long-term regulation in the DNA as well, which is related to like our growth hormone. So both of these mechanisms I'll discuss later. But before going into details, as I said, like when cyclic AMP binds with the inactive state, it releases the active moiety and then it targets a protein and attach a phosphate group to that protein. So what happens is kinases mainly targets the serine or threonine amino acid of a protein structure, and very rarely it also targets tyrosine kinases too. So it binds a covalent bond to that covalent bond between the phosphate group and the amino acid. And why serine, threonine, and tyrosine? It mostly targets the amino acid that has a hydroxyl group at the end. That's why serine, threonine, and tyrosine, all these three has a common hydroxyl group in the structure. So serine kinase is a protein kinase A. So upon activation, it targets this hydroxyl moiety, transfers one phosphate group from ATP to the hydroxyl in the place of hydrogen, and then it is an active stage, and this phosphorylated serine then go on and perform its enzymatic activity in the cell. Usually the target of protein kinase A is presented like this. RR is the regulatory subunit and it's very tissue specific. Like this portion of the amino acid sequence is based on which tissue we are looking into. S is a serine and T is for threonine, and this X is like the variables. Like this does not has anything to do with the protein kinase binding or the regulatory function. So this is the basic target site of protein kinase A, and this is how binding of CAMP regulates the activation and inactivation of protein kinase A. So remember in the earlier photo I said like it's a tetrameric structure. So this is how a protein kinase A is usually in the cell in an active state. And the blue dots here are the CAMP, and the green portion is the regulatory subunit where CAMP will bind, and it is usually the target of any drug. And the red one is the catalytic subunit, which is the active unit. When it is released, it will bind. It will take a phosphate group and bind it to a protein. So what happens is when the concentration of intracellular calcium is increasing, it will bind to this portion. Like I marked it like 1, 2. So for 1, 2, 3, 4, for each protein kinase A to get activated, it needs 4 CAMP to bind, and this binding is kind of like self-catalyzing, meaning when one CAMP molecule binds to this place, it will increase the affinity of another binding site to look for another CAMP moiety to bind with it. So it's like when one is, it's again an allosteric conformation changes. To simply put, when one CAMP is at its place, it will make sure the second one is there faster than itself, and so on and so on. And when all the CAMP is attached to that portion, the catalytic subunit will be released and it will perform its function. But then again, what happens is you get asked here, like, okay, if CAMP is here, and then this should always stay on, but it doesn't happen, because as soon as all the 4th site is full with CAMP, it will lose the sensitivity towards CAMP, and then all the 4 of the CAMP molecule will be released, and it will again find its catalytic subunit and go on into its deactivated state. If anything happens in this particular mechanism of action, the PK will be stay active for longer time, and hence the reaction will stay active for longer period of time. But that doesn't happen because it's happen normally. In a pathological condition it can, but normally what happens is CAMP will cause the inactive tetramer to be active form, and then again by the nature of its binding it will release itself from the cell and become inactivated, and then CAMP will be converted into 5-AMP by phosphodiesterase enzyme. So this is how CAMP actually regulates PK, and it is very important for PK activation. Now previously I mentioned that PK performs short-term and long-term regulatory control in our body. So this is an example of a short-term. I know there are a lot of stuff in this slide, so I will just walk you through one by one. So this is a phosphorylase B enzyme which is responsible for glycogen breakdown. It converts glycogen into glucose form and releases it into blood. So phosphorylase B is an inactive form, and the active form is called phosphorylase A, and it can become active in two different pathways, and it can become active with or without the phosphorylation. So first let's talk about this portion because it's more relevant to pKa. So this is an inactive T form. T means tensed form. Like the structure here, as you can see, is different. And when phosphorylase kinase causes a phosphate group from ATP to bind with it, it becomes phosphorylase A from phosphorylase B. See, these phosphate groups are binded, but it's still in a tensed form. And when it is here, it automatically changes conformation to become this R form, which is called relaxed form. And this then go on and perform its activity like transforming glucose from glycogen and hormone. So this is a pathway that happens when there is an extracellular influence, like a ligand will bind to a G protein, and then it will increase the intracellular CAM concentration, and then it will activate pKa, and then the pKa will convert phosphorylase B from B to phosphorylase A, and then it will perform its function. So this is one pathway that glycogen and glucose is regulated in the cell. Another pathway is like independent of any extracellular influence. It responds to intracellular condition, which is allosteric effectors, and it does not work through the phosphorylated activation pathway. It happens when there is like metabolic sufficiency, like let me tell you about the hunger first. Like when we are hungry, the 5-AMP will be activated and it will convert the inactive tense form to active R form without any phosphorylation, and it also has its own activity inside the cell. And when we have like enough food, there is a metabolic sufficiency signal. It will then convert the active R form with the help of ATP and glucose 6-phosphate to inactive T form. So the whole point of this figure was to just just to give an idea about how phosphorylase A and B enzyme functions with or without the phosphorylation. But when there is a phosphorylation, it means there is an extracellular influence, like a ligand has bind to G protein and pKa is involved. Now why they discuss this in very details is because they go into this mechanism of action, and to understand this, the previous one was, I guess, important. So this is a general mechanism how protein kinase A functions inside the cell. So in liver cell there is a beta adrenergic receptor, like each cell has 100,000 of it. Not all the cell needs to be occupied for PKA to exert its activity. So what happens is when adrenaline binds with adenylyl cyclase, it increases the cyclic AMP concentration inside the cell, and this CAMP will increase concentration of CAMP will activate protein kinase A. Now protein kinase A will then activate phosphorylase kinase, which will convert phosphorylase B to its active form, which is phosphorylase A. Here is the additive phosphate group. Now the red marks the active stage of that enzyme, and that enzyme then will convert glycogen to glucose 1-phosphate, which will be released into the blood. Now this protein kinase A, where in one hand is activating this pathway, it is also inactivating this glycogen synthase A. By adding a phosphate group, it is inactivating. Like I told you before, like pKa will perform this contrasting function of activating and inactivating enzyme. So when glycogen synthase A is inactivated by adding a phosphorylate group, it will stop making glycogen from the glucose. So that is how pKa kind of controls the blood glucose and glycogen levels. So what happens is, here you can see that adrenaline, when it is binding to adenine cyclase, it is like very low concentration, like 0.1 nanomole per liter, but it is releasing a huge amount of glucose in the blood. So the benefit of this protein kinase A pathway is it can take a very weak signal and then can amplify it into a large signal or large end product. But the problem is that, as you can see, it takes a lot of steps to release the glucose. So it is slow, but it can amplify signal, as opposed to what we have discussed last 2 weeks, like calcium channel, which was very fast. That was like a recurring thing. The calcium channel activity is very fast, but it's slower than that. And this is a long-term regulation of pKa. Other than regulating glucose, it can also regulate the transcriptional factors in the DNA. Now it happens in two pathways. So one is like calcium modulated pathway with IP3, galvadine, CAM kinase 2. But for pKa, this pathway is more relevant. Like when a ligand binds to G protein, ATP converts into CAM, and CAM activates pKa. pKa then goes inside the cell, and then it phosphorylates CRAB. And upon phosphorylation, CRAB forms a dimer. Now this dimer then goes on as a transcriptional factor and target the response element. Like it has a specific target site on the DNA, which is called CRE, and which is CAM response element, and it binds with the DNA, and then it like starts transcription of a process. And once its activity is done, phosphatase comes inside the cell, it then dephosphorylates this CRAB, and then it becomes monomer again, and then the balance is restored. Anything that hampers this process will cause genetic defects or any disease later in the life. Like this whole process of activation of CRAB dimer by pKa will take 15 to 20 minutes, and then with time, like 4 to 6 hours, it will dephosphorylated and release into the blood again. That's like the normal activity. But if anything happens to this phosphatase, like ocadic acid is a substrate that will block the phosphatase, then this transcription will not stop, and it will keep on doing whatever it is doing. So this is like sometimes this pathway is taken advantages of to treat some disease, and sometimes some pathology will use this pathway to like make sure some transcription is on and on for longer time. So this is the long-term pathway of pKa. Now pKa and activation of ARC has been a question for scientists for longer of time. So initially it was like discovered that pKa works downstream of CAM. It is a target for GS protein. So when ligands bind to GS, it causes like intercellular CAM concentration to go up, which activates pKa, and then it binds with substrate and blah blah blah, what I just told earlier. So this pathway has been established fast. But then scientists discovered that pKa also functions to the GI with the GI protein also, but they didn't understand how. Later they found out that when pKa is activated, it not only attach a phosphate group to a target protein, it also causes conformational changes into the G protein, and then shifts its function from GS to GI, and then initiate this pathway also by targeting beta, gamma, RAS, and R. Here is a photo that will like it's an easier version, I would say, this figure. So when GS is activated, it activates pKa. Now pKa goes to the G protein itself and binds the phosphate group there, which converts the GS into a GI protein. and then it starts this ARC pathway. But it, with the help of a phosphorylase enzyme, it again gets like dephosphorylate, and then it comes back to its original state as a GX. So this has been a very important discovery of pKa. Like usually it has been thought that pKa works downstream of G protein, but then for GI it works upstream of it. So now I have been talking about CAMP and pKa a lot. The chapter at the end also highlighted the activity of CAMP, and it doesn't always require pKa for its activation, because as I have discussed before, like CAMP pKa pathway, although powerful, is a bit slower. So sometimes in some cell, CAMP performs activity without the help of pKa enzyme, and one of them is olfactory cell signaling, where the scientists discovered iron-gated channel, which is a CAMP-gated ion channel. Here, when the ligand, it is actually an odorant, binds with the G protein couple receptor, it converts like GTP to cGMP, and this cGMP will binds to this potassium channel, which causes the potassium to go outside of the cell. This hyperpolarization will activate this CAMP-gated ion channel that will cause sodium ion to come inside, which causes depolarization, and it will open the calcium voltage-gated calcium channel, and the calcium ion will come inside the cell, and it will like in turn transfer the odorant signal to the brain. So this is a pathway where CAMP does not require pKa to perform its activity, because it is faster than the regular CAMP pKa pathway. Another action of CAMP, which is independent of pKa, is inside the cell, like RAP1 pathway. Here, this is the plasma membrane pathway, and this is the organelle membrane, like outside of mitochondria or outside of endoplasmic reticulum. This pathway takes place, which is completely opposite of what happens on the on the plasma membrane. So when CAMP binds with exchange protein directly activated by CAMP, that's like EPAC. So when CAMP binds with EPAC, it converts RAP1 GT. Sorry, RAP1 is a monomeric GTPase, so it converts RAP1 to GDP, and so the pathway is what happens in the plasma membrane and what happens in the organelle membrane is same. The difference is between SOS protein and EPAC protein. In the presence of EPAC and RAP1, the organelle membrane has an antagonizing action of the plasma membrane. The reason the chapter defines it, because it happens like that, it kind of maintains a balance in the cell. When some some signal is coming outside of the plasma membrane and it has performed this action, and the cell needs to go back to its original state, then the CAMP that binds with the external plasma membrane can also bind with the organelle membrane, and it can kind of antagonize that action and brings the cell back to its original state. So this is so far their explanation from it, but they have also considered the limitation, like it still needs to be like looked into, especially like the function of RAP1 GDP in the organelle membrane and how this antagonizing action can be utilized in a disease condition. So from major takeaway about the pK, if from the whole presentation, is that it adds a phosphate group to the protein. It is a tetrameric enzyme that requires intracellular CAMP to be activated, and pKa and phosphatase works as a yin and yang to maintain the harmony of cellular function. Dysfunction in any one of these enzymes will lead to a disease condition. Now for my portion, the paper I found is sorafenib for the treatment of hepatocellular carcinoma. So now this paper focus on the mechanism of sorafenib and two global random clinical trial that established sorafenib as a treatment of hepatocellular carcinoma. So sorafenib is an oral multi-kinase inhibitor because it inhibits multiple kinase in the cell, and it targets RAF kinases and receptor tyrosine kinases. This is the structure of it, and this is its general inhibitory profile. So as you can see, there is a whole lot of kinase it targets, and this is the IC50 value of that target. So since it targets multiple kinase, it calls a multi-kinase inhibitor, and its anti-tumor activity mainly it targets tumor angiogenesis and also proliferation. It causes apoptosis and anti-angiogenesis action in the cell. So this is a very busy slide where they try to establish the mechanism of action and then the target for sorafenib. So this is a tumor cell, and this is the vascular endothelial cell. Tumor cell will shoot out vascular endothelial factor that will cause formation of vasculature around the tumor to grow more. So first let's see what we have here. So it is a TGF alpha when bind to EGFR to RAS, RAF, MAP, ARC pathway. There is a survival or proliferation pathway that the tumor cell utilizes. So SF is a sorafenib. In the RAF one also inhibits the mitochondria to kill the cell. So sorafenib inhibits this inhibitory action and causes this autocrine loop to stop and causes cell apoptosis. On the other hand, a tumor cell will shoot out factors like PDGF, VEGF, which is like platelet-derived growth factor receptor, and vascular endothelial growth factor receptor, which will then cause like angiogenesis, differentiation, and proliferation, and all sorts of function. So sorafenib kind of also acts on those also. So when these PDGF and VEGF factors are shoot out by this tumor cell, it binds with this beta PDGR beta receptor and VEGRF2 receptor, and also with RAS, RAF, MAC pathway. It will cause angiogenesis, differentiation, and blah blah. And also like this cell here, RAF also inhibits the mitochondrial induced cell apoptosis, and sorafenib induces this pathway, and also it induces the initiation of RAS RAF pathway as well. So not only it is causing the solid tumor cell to die, but it is also preventing angiogenesis and the tumor cell to grow. So this has been like a fantastic option for hepatocellular carcinoma. So first I will go with the cellular result that we got, in vitro result, and then I will focus shortly on the clinical trials. So these are the two types of cell that can mimic the hepatocellular carcinoma in vitro. In PLC PRF5 cells, as you can see, the sorafenib with the sorafenib has an inhibitory effect in a dose-dependent manner. The IC50 is quite high, but it does kill the cells in vitro, and same with Hep G2 cells. Here the IC50 is kind of similar to that, and this is like the DNA fragmentation, like whether the cell is actually destroyed and the DNA material is outside of the cell or not. So kind of similar for the two HCC cell model in vitro. So basically it inhibits the cell proliferation and apoptosis in the HCC cells. And this is the Western blot where they concluded that it targets the RAF-MAC pathway, but unfortunately I could not find any graph of this analysis. They just posted the photos of Western blots. So this is PLC PRF1 cell. This is diamesonabakil. This is sorafenib, and this is I think this is a positive control, like a known inhibitor of this pathway. And this is their Western blot analysis, but since there was no graph, I cannot actually tell the percentage of inhibition. But yeah, that's.
But it looks pretty good, especially in those
higher,
cells, yeah.
Higher concentration. It kind of.
Yeah.
Works. So these are like the two cell model of HCC.
Yeah.
In both of them, sorafenib was effective in.
Isn't it funny? The
Yeah.
one cell type, look how nice the westerns turn out, and then the other cell type, it's all like,
All fuzzy.
yeah, isn't that crazy? Like, what is that? Why does that happen? Why some cells is all fuzzy? That's interesting. I'm assuming they use the same antibodies, the same protocol, right?
Yeah, yeah.
Yes.
It's technical issue, I think. That's what I have experienced. Like,
So you think two
Something.
different people did these, maybe?
maybe one time the person is doing it in a very ideal way, otherwise the next time it's not that. Like, because it's a long procedure and it can have different, like, the steps like transfer, there is a problem with the transfer,
Yeah.
or maybe the transfer is not that good, or maybe, I don't know, like,
I don't know, but I mean, they show their best. You show your best plots in the
Yes.
paper.
Yes.
but the cell type is also different, like,
That's what I'm saying.
And
Why
she's
in different
doing-
cell types? Why in one cell type do you get these fuzzy westerns, and then in another cell type, it's pretty crazy to think about it. Anyway.
there's a good difference between, like, that one is really good.
Yes,
that
right.
one
There's
is
a
really,
real, I mean, that's really interesting to see side by side, especially that first plot in the Hep G2 cells.
Yeah.
That one's not very good at all
Yeah.
compared to what. Look at the ones with the
Yeah,
PLC. What's that? PRF.
yeah.
Five
PLC, PRF 5 cells.
Yeah.
Yeah, but I kind of wish they would present it in a graphic
Yeah,
format
would've-
so that we would have an
Excuse me.
idea.
It would have been nice. I mean, it's pretty obvious that there's inhibition of. Although, in those cells, right, in that first with the 0.1, it looks like it was almost activated phosphomec, but
But then
compared to DMSO, right? Yeah. Yeah.
ma'am, should with the when we
Can you go back a second?
yeah, sure.
when we basically calculate the numbers, the signals of the phospho arc and the normal arc, when there is more phospho arc, the normal arc should go down, or it shouldn't be.
When there's more phospho, or, I mean, so if you're saying total of the total, or yeah, but it doesn't. You know, it often doesn't go down the total concentration. It's just the percentage of the ERK that's being phosphorylated increases.
Yeah, like I what I mean is that if the arc is phosphorylated, the normal arc should go down because it's phosphorylated, or
It
maybe it's
should, but when you do normal, or if you're saying non-phosphorylated,
non-phosphorylated. Yeah.
that takes in non-phospho. That's total ERK, so it's non-phospho plus phospho.
Both.
Right. So it doesn't necessarily have to go down because I think what it is just a percentage of that total ERK is more of it's being phosphorylated.
But it might not be like same for every other protein. Like
No,
yeah,
it might not be, right?
for example, in our lab, like we do alpha-synuclein, total alpha-synuclein and phospho CD1 to 9.
Uh-huh.
So when we do it, like most of the time when phospho is increased, the total is very light.
Yeah, okay. That's interesting. So does the total antibody not recognize the phospho and the non-phospho, or does it just, or is there actually a decrease in the, so maybe there's two things happening, a decrease in total protein and an increase in the amount of that total protein that's becoming phosphorylated.
Yeah, but there is one difference. Like in that phosphorylation, it's also protein post-translation modification as well. So that's why maybe the total protein, the total alpha-synuclein antibody is not recognizing.
Recognizing, yes.
Yeah, maybe
right.
that would be the
could be
reason.
possible,
That
yeah.
would be the-
And I'm just also looking at that AKT there. Oh, that second lane with AKT is actually, they added growth factor. I see that, okay. And why did they do AKT in the other cells? I'm always curious. I'm wondering if like it just didn't show what they wanted it to show. You know what I mean?
Yeah.
Like if you show all that in one cell type, why wouldn't you show that in the other cell type? But yeah, anyway.
So the point of this question block was to establish that okay, this sorafenib is working through RAF-MAC-ARC pathway, like blocking that pathway and causing cell apoptosis. But then in the previous slide, you also saw that it also inhibits the binding of the different platelet and growth platelet factors and growth factors to its receptor. So it kind of shows that it also causes, it also inhibits angiogenesis and cell death, independence of that MAC-ARC pathway. So it inhibits the phosphorylation of this EIF4E factor, which is a transcription factor. When phosphorylated, it will transcribe many cellular components. So it inhibits the phosphorylation of that, and also MCL1 is an anti-apoptotic factor. It also inhibits that and causes cell death. So the point of this slide was to establish sorafenib works into a pathway. It blocks the MAC-ARC pathway, but it also works independent of that. But then again, it did not show a graph of this analysis, so we cannot conclude the statistical analysis and so on. And this is immunohistochemistry assay to show that it actually blocks the angiogenesis. This presented the microvessel density and the amount of phosphorylation of ARC. And apoptosis in vitro, this is like CD34 phosphorylated ARC, and this is the tunnel protein. But for this presentation, they did present.
Let's see, what's that? What are we looking at there?
So this is the
tunnel,
immunohistochemistry of the tumor cells to actually show like it is preventing the phosphorylation of ARC, and it is like reducing the angiogenesis.
Oh, that's the microvessel. Okay, I see. Yeah.
Yeah,
Okay.
they did get a dose-dependent statistical significance.
Yeah.
And this is the animal study where they showed that sorafenib against. So the tumor cell we discussed, it was grafted into the mice model that was immunocompromised, and then they measured the tumor weight and day post implantation. So the round one is untreated, the star is baking, sorafenib is this one, the block circle, and this one. This is the lower dose 10 milligram per kg, and this is 30 milligram per kg.
Wow.
So at higher doses, the tumor did not grow
That's
at
amazing.
all. And this is the other type of cell that we discussed that is also implanted. Oh, sorry, no. This is the one type of cell, like PLC-PRF5 cell. This is 16 days post implantation, and this is 21 days. So yeah.
Wow, that's very effective.
And here they introduce higher dose, like 100 milligram per kg, and there they saw a shrinking.
Yeah, right? Wow, that's amazing.
Yeah. And these are the landmark clinical trial outcomes that establish it as a treatment. So this is the probability of survival, and this is like marked since randomization. So sorafenib and placebo group, as you can see in the placebo group, the probability of survival get lowered with time, where sorafenib did not. I mean, this is not a positive slide, but then again,
No.
it kind of not decreasing the mortality rate, but I would say increasing the lifespan of the cancer patient. And this is the probability of radiologic progression, like whether the patient would need radiotherapy at the end of their cancer journey. So sorafenib kind of lowered that probability as well. So these
I'm
are,
surprised it's not better, considering what they saw in the mice.
yeah.
I mean, it really knocked out those tumors, so I'm guessing, yeah.
I guess they kind of jacked up with very high doses in the mice. With human, I don't think that's
Yeah, right.
possible
That's
without
true.
avoiding any side
Yeah,
effects.
that's true.
So these are the side effect profile. So these are the two major studies. One is SHARP and one is Asia Pacific study. So the major side effect was hand-foot skin reaction. Sorafenib was 8% and 10% in two studies, and then diarrhea, hyperbilirubinemia, fatigue, weight loss, and hypertension. So overall, they're
Not so bad.
yeah, not so bad, I would say, but there are side effects.
I mean, hypertension's bad, but I mean, weight loss and fatigue, that's pretty normal with cancer treatments. Hand, foot, skin reaction, I mean, depending on how severe it is, it could be pretty easily taken care of. Diarrhea stinks, but it's not. Hopefully, it's not so bad that they'll become dehydrated if they're drinking water. No, it's not so bad.
So this is my presentation on protein kinase. I hope I was.
Yeah, no, it was really good. Thank you so much. Good job.
Right.
Anyone have any questions for Oksana?
So,
Yeah.
are there any resistance for the sorafenib?
Not yet, no. But the thing is with this kind of drug, what happens is in phase 4 trial, they don't always see the similar efficacy as they saw in phase 3 and phase 2.
Right.
So there are varying degree of efficacy that has been reported, but it's still one of the most prominent prescribing drugs.
Do you know if the population of the sharp study was in Western? Because I just noticed
The
there's
sharp
Asia
one
Pacific.
is Western. The Asia Pacific has, I think, 5 to 7 countries.
Eastern
Population.
population.
That, yeah, good.
Mm.
I
Eastern
mean, that's
cultures.
good that they did that.
Yeah,
Yeah.
that's interesting. And it looked like it worked in both populations, so that's good. That's good news.
I have a question related to one cycle you showed where PKA was going like working upstream.
Oh, right, yeah.
Yeah, like can you tell me that which what's the role of CAMP over here? Like, is there any role of CAMP over here, and how
Yes.
it's functioning? Because it's a cycle. So, what is the end product and how it will be functioning? Yeah.
So this is ordinary G protein, and upon binding with ligand, it releases intracellular CAMP, which activates PKA, and then it has its regular function. But sometimes this activated PKA, it adds a phosphate group to the G protein itself, which changes the conformation of the G protein and converts it
Into
to a
GI.
GI protein. And when a GI protein it is converted GI protein, it will bind to a different ligand, and then it will start another pathway. So the role of PKA here is for GS it is working downstream, and then for GI, some GI, it is working as an upstream activator.
Like my question is like, what benefit will be getting like with this cycle? Like again, you said that the ERK pathway will also activate GS again, and then
Right.
GS again will activate PKA. PKA again will convert GS into GI,
Yeah.
and then it will again go to ERK. So like, what is the benefit of this cycle overall? Like,
I think sometimes scientists don't understand why some pathway is still activated. Like, it's not supposed to be activated. They thought that this protein is GS and this should only work to this pathway, but why it's working to that pathway? I think discovery of this kind of give them an idea like
Switch,
single
yeah.
protein can switch between GS and GI and can totally perform antagonizing actions in the cell.
so you
And
will get
I think that switch to GI is like a negative feedback onto the PKA, right? Because that shuts down or decreases cyclic AMP production. So then but then the ERK becomes activated. So it's probably a shift in what's needed for the cell too, you know? What kind of proteins need to be upregulated and such.
and it's in this cycle the CMP is not playing any role. Like when
No, so
PKA is going
yeah.
upstream.
It's like when PKA is activated, CAMP has no role in. CAMP
Right.
just activated the PKA and it is going downstream of GS and then upstream of GI.
Right.
That's the whole point of the whole point of showing it was that PKA can work both ways, upstream and downstream.
Right. And PK is kind of self-regulating itself in that way. We're switching to GI, right? Yeah, it is interesting. Yeah, very interesting. Well, so much to think about. You all are doing such a great job, and this class definitely, I think, what I like about this class is we don't necessarily come up with a lot of answers, but we come up with new mechanisms that we can kind of apply to all of our research. And so,
Yeah.
yeah. Okay, I really have to run. Thank you.
Thank you.